In the previous chapters, we learned how to identify and clone a gene from various organisms. In this chapter, we will try to apply that knowledge in order to produce the protein encoded by our gene of interest. This means that we will try to produce the maximum amount of protein during the minimum amount of time for the minimum cost, which is really what biotechnology is about. In this field, there is a lot of engineering decisions and optimizations that need to be considered. Take a step back and look at our proposition to produce proteins from cloned genes. This is a part of, or can be seen as a part of the larger initiative, sometimes called the green economy, where we would like to replace traditional chemical industry, where we produce products from fossil raw materials such as oil, using high temperature and high pressure, where we also produce lots of byproducts. Instead, we would like to use renewable streams of substrate, such as waste, a cell factory instead of a chemical factory, where we use the low temperature and pressure that's associated with biology to produce a big part of product. The process is thought to be more specific, and because of the specificity of biological catalysts, the amount of byproducts is thought to be little. The disadvantage of the bioproduct is that we usually get a very dilute product, and we need to often remove water because biology happens in dilute water solutions. There are many different products produced nowadays from industrial fermentation. Notable examples are penicillins, that is produced by the mold penicillin. Of course, the most common bioprocess is perhaps the production of alcohol using Saccharomyces cerevisiae. But we also have in this list a couple of enzymes, invertase, that are used for production of high fructose corn syrup using Saccharomyces cerevisiae. Although these are interesting, all of this, we will focus mainly on how to produce enzymes using a couple of industrial protein production organisms. If we look at the process economics of biotechnology, we can see that it's controlled to a large degree by the value of the product. And the value is often high because there is just no other way to make some of the products. If you look at the selection or categorization of products produced in biotechnology, primary metabolites such as ethanol and maybe oil is quite a low value product, and here the design of the process become very important. Secondary metabolites would be, for instance, antibiotics. Penicillin is a good example. These are pricier, so we can afford a more expensive bioprocess. Enzymes are very valuable depending on what they are. This is actually a very heterogeneous group. Therapeutic proteins that are used to treat or diagnose disease are the very most valuable. Although enzymes in general, there are very specialized high-value enzymes, but there are also enzymes that go into dishwasher detergent that are produced on a massive scale. But I think we can say anyway that enzymes and proteins are probably the most valuable products that we can do in biotechnology. A little because these products cannot be made in any other way. There is no process that competes with this. There are several different strategies to cultivate microorganisms in general. Two notable examples are batch culture or closed culture, where we simply prepare a quantity of medium, that is, with all the nutrients that we need. We inoculate it with a small amount of cells, and these cells will divide and produce our product. And then, depending on what we want, if our product is in the supernatant here after centrifugation, or inside the cells, we either continue to process the medium or the cell mass. Here, I already mentioned the problem of dilute solution, and this is something we will always have to deal with. Most of what we deal with is actually not cells, it's the medium. Most of the system is water, really. We also have continuous culture, where we feed at a steady state fresh medium. We have a way to stir this. We also have to stir this, by the way. They just didn't add it to the cartoon. And then we take away an equal amount or an equal stream as the input. So the volume here stays the same. This means that we can control the productivity quite easily by changing the speed at which medium goes in and the medium and cells go out. This kind of culture is called continuous culture. There is also one where you actually start with a small volume. You don't have any exit, and you add medium inside at a low rate, and then you fill up the fermenter slowly. This is called fed batch. And there are different reasons to choose any of these. We can say that this continuous culture is roughly 10 times as productive as the batch culture. But in practice, we rarely see continuous culture because it's difficult to control the sterility of the medium. And if you have a contamination, it's difficult to deal with. In the batch culture, you prepare everything new every time you grow the cells, so it's a lot easier to have a consistent product. I would say that one problem of the batch culture is that you have to have all the substrate in the medium that is needed during the cultivation, whereas in the continuous culture in the fed batch, you add the substrate slowly. This is significant if your cells, and they usually are regulated due to the substrate that they sense, and this can affect the process in very complex ways that you have to deal with. One question would be, where does genetic engineering come into all this? I mean, the cell already has the gene that we want. We could simply take the original cell that contains the gene for the protein that we want and put that in our fermenter and grow it. The problem is that it might not be so easy to grow the cell. If we, for instance, want to produce a human protein from a human gene, it's impossible. You can't extract protein from human bodies very easily. So the solution is to take the gene and put it in another organism that is easier or more economical to grow. In this case, we have to deal with three things. We need to adapt the regulatory sequences before and after the gene, because usually the promoter that comes with the gene is not going to work in the new host unless the hosts are very, very similar. We also need to make sure that the ribosome binding site, this is important for the subsequent translation of the protein, it needs to be adapted for the host organism and not the organism from where the gene comes. And then we have to have an efficient terminator that terminates the RNA transcript in order to have efficient translation. Sometimes we can get away with producing proteins from different bacteria in another bacteria and different eukaryotes in another eukaryote, but between prokaryotes and eukaryotes the control sequences are very, very different. The E. coli promoter tend to be short, only about 40-50 base pairs. The animal or yeast or eukaryotic promoter in general tend to be much longer, several hundred base pairs, and much, much less defined where these transcriptional signals that are important, where they are located. One similarity is this Tata box or the Pribnow box in the animal. So the solution is simply do away with everything except the exact coding sequence of the gene. So we could combine any promoter and any terminator and any gene as we like, but the most common method is to choose an expression vector that has a certain promoter, a certain ribosome binding site, a certain terminator already cloned inside. So the only thing we have to do is put our coding gene, our protein coding sequence here in this unique restriction site, and hope that we will get a protein. There's usually a large library of these vectors, and they all differ a little bit in the promoter or in other signals, and also copy number is usually a good way to engineer different levels of expression. The question is here, why would we want to engineer a different expression level? Shouldn't we always want the absolute maximum? Here we see an example. We pick a strong promoter, and the strong promoter means we get lots of transcripts, lots of mRNAs. All of these mRNAs goes to the ribosomes and we get lots of proteins. And if we have a weak one, we get less of both transcripts and proteins. Well, the problem is that if you express too much, and perhaps in the different or in the wrong part of the cell growth phase, you can get too much proteins. So the proteins will aggregate together, and or they will have some toxic effect on the cell. So in order to achieve the absolute optimum of protein productivity, that is the maximum amount of protein per time, per unit volume of reactor, and per amount of substrate, you need to balance the two effects. Not all proteins have a toxic effect, but many do. So there are various ways of dealing with this. A popular way to optimize the expression level of a gene is to use an inducible or a repressible promoter. That is a gene that is normally off and that can be turned on by a regulatory chemical. It's an inducible gene. One that is normally on but can be turned off is a repressible gene. And using these, especially if the system allows fine tuning, we can add more or less of these regulatory chemicals in order to achieve a balanced amount of mRNA. And this can help us to achieve this optimum where we get protein but we don't affect the growth too much. We can also completely separate the growth phase and the protein production phase. We We can grow the cells completely with no transcription in this case, and when we have a lot of biomass in the end, when the cells no longer grow, we can add our regulatory chemical or take it away. It's easier to add, of course, and we get the protein production. This slide shows some different examples of regulated protein production systems available for E. coli. The first example is, of course, the lac promoter, which is the classical example of gene regulation in biology, where a derivative, in this case IPTG, a derivative of lactose, can interact with the lac repressor and turn on or off the transcription of these genes. Another, maybe less known example is the TRIP promoter. It normally controls the expression of the TRIP gene. This can be induced by 3-beta-indole acrylic acid, and it can be repressed by tryptophan. This is obvious or easy to understand, since if we have tryptophan in the medium, there's no need to make more tryptophan, so adding tryptophan turns off the promoter. The TAC promoter is a combination of the lac and the TRIP that is also widely used. It's repressed with IPTG, or induced, sorry, it's induced with IPTG, just like the lac promoter is. There are also examples of temperature sensitive promoters, where if we grow at a low temperature there will be little or no transcription, and if we raise the temperature there will be activation of the promoter. This promoter is from the lambda phage, so it usually controls the lytic versus lysogenic cycles of the phage. In practice, a very widely used system is the T7 promoter. This is a different promoter because it's actually not an E. coli promoter, it's a promoter from bacteriophage T7, and this has been heavily modified so that it's can be induced by IPTG, just like the lac promoter, but the induction of the induction is actually for producing an RNA polymerase that is has affinity for this promoter that will then create transcripts. This system is so widely used that I will talk a little bit more about the details of the T7 system. The T7 system, or the PET system, has a very ingenious way, two-step way of controlling transcription of our target gene. We normally clone the target gene in a plasmid, just like in many other systems. This plasmid has a T7 promoter, and it also has a place for the lac repressor here. So what we don't have in all E. coli is this T7 polymerase. So we need a E. coli that is lysogenious for the DE3 phage. This is a phage that is incorporated into the genome, and it has the T7 gene 1 that encodes the T7 RNA polymerase. This gene here is in turn controlled by the lac repressor, the lac gene promoter. So what we have is also a gene, third gene here, that is the lac repressor, that encodes the protein that actually represses the promoter. So without any induction, we cloned our gene, we have everything together in the cell. This repressor will prevent the production of this RNA polymerase, hence there will be no production of our protein. But if we add IPTG. It will interact with the repressor and allow the RNA polymerase to produce this RNA polymerase. So we have a two-step induction. And this RNA polymerase will attach here, and this lac repressor here will also be inactivated because of the IPTG added, or some other lactose analog. And this will start transcribing our gene, and the normal protein production machinery of the cell will then produce the protein. So why is this system so popular? We can say that because of this two-step induction, the difference between induced and non-induced, the induced and non-induced levels are very high. It's almost zero when not induced, and it's very high when it's induced. We can ask why is it so high then? It happens so that this polymerase has a very high affinity for the promoter, much higher than normal, because this usually works only in a phage, and I suppose the fine-tuned regulation is not necessary. It's also so that this RNA polymerase only recognizes this promoter. So all of these polymerases will make transcripts from this protein and not all the other genes. If we compare with the E. coli RNA polymerase, has many other genes to attend to. But here we have a very focused production. So we have enormous amounts of protein potentially being produced from this system. Sometimes we can correctly clone a gene into an expression plasmid, and we still don't get any expression, we don't get any protein. Often this is unknown, unfortunately, the reasons for this, but there are some suggestions that some sequences of the mRNA, like here, can form strange structures that will obscure the ribosome binding site. This one needs to be accessible to the ribosome in order for the gene to be expressed. We can solve this. This might be difficult to actually do something about, because it might depend on the actual sequence of the actual gene here, and we don't have we can't control that. Maybe we see here this blue box here represent the gene sequence that we actually want, so we don't want to change this really. What we can do is we can take this, our sequence, and put it inside another gene of E. coli. This means that we get a little bit of the gene of an E. coli gene expressed before our gene. This may or may not be a problem, depending on what our immediate goal is. We need to, of course, take care that the fusion, the reading frame here is the same, because the ribosome starts translating in this part of the mRNA, and we need to carefully attach our recombinant gene in the rate, so that the triplets of the are preserved of the E. coli gene here. If we fuse incorrectly, we will get a nonsense transcript, and we will not get any activity. As I said, in the end we will get a fusion protein, so a little bit of E. coli here, and the bigger part here will be the protein that we want. This is perhaps not a big problem. If this is an enzyme, it might work, might be effective anyway. If it is a therapeutic protein or something more clinical, this might be a problem. This segment might provoke immunological reactions, for instance, and you might not want to leave it on. Another reason to actually make a fusion protein might be to detect or to purify, facilitate purification of the protein. In this example here, we make a fusion of our protein or our gene, and the gene then coding glutathione S transferase, which is a protein that has a high affinity for a molecule called glutathione, which is really a small protein with 3 amino acids: glutamate, cysteine, and glycine. And we can use small beads of agarose gel, small balls of agarose gel, and we can attach these glutathione molecules on the surface quite easily. When we have fused our protein with a glutathione S transferase, they will stick very strongly to this glutathione on the surface. So if we pass our cell extract or something containing our protein, they will stick to the beads, and we can wash all contaminants, and our protein will still be stuck to our beads. We can then add more glutathione in soluble form so that we outcompete these beads, and we can dissolve the protein. It will be a pure fusion protein here, containing quite a bit of glutathione, of course, but not much else. The glutathione and the fusion protein. This glutathione S transferase is a detoxification protein, really, and this glutathione in the cell is used to detoxify certain toxic molecules. So the glutathione S transferase picks one of these molecules, pick the other molecule, whatever it is that is dangerous, and attaches this to make it less dangerous. So it's quite efficiently to bind to this molecule. Even so, we might not be able to express our protein in E. coli. One common problem is that proteins that we try to express end up in inclusion bodies, that is, insoluble aggregates of our protein inside the cell. And this might just be a property of the protein itself. It's perhaps incompatible with the environment inside the E. coli cell. Perhaps we try to express too much of it, and there's just too much, and they crystallize. This may not be the end of the world. We can quite easily purify these inclusion bodies, and if the protein allows it, we can try to renature the protein from the inclusion bodies. This depends again very much on the protein. This is not always successful. Other than that, there are, of course, other kinds of problems that we can have by trying to express. The most obvious one is the presence of introns. We usually take care of this in the cloning step by cloning only the coding sequence. We use, for instance, a cDNA instead of the actually genomic DNA. We can also find that some codons inside our gene resemble stop codons for the E. coli. And although the genetic code is universal, there are slight differences and slight preferences. And we can actually, what can actually occur is that it simply stops the mRNA in the middle here. And we've already talked about codon bias, how some codons, for instance, here in this table, we can see that E. coli have a strong preference for CGC, for arginine, and perhaps, let me see, if you compare that to human, if you try to express a human gene, all of these codons here might occur quite often, but they're very rare in E. coli. And this means that the protein production, the ribosome, is sitting and waiting for the right transfer RNA, because there's not a lot of these transfer RNAs being expressed, perhaps. We can solve this. We have already talked about making synthetic genes where we maintain the sequence of amino acids, but we switch the codons to be more similar to the ones that we have in our host organism. If despite all our efforts we cannot manage to express our protein in E. coli, there are a couple of other more complex systems that we can choose from. The disadvantage of these systems is that cost and complexity tend to rise when you choose something else than E. coli. So you normally do this when E. coli fails. The first choice might be yeasts and fungi. These are simple organisms still. The genome of the Saccharomyces cerevisiae, the baker's yeast, is only 3 times that of E. coli, so it's not so much more complex. There are other yeasts, such as Pichia pastoris, that have other advantages. There is filamentous fungi, like Aspergillus nidelans and Trichoderma rhizae, that each of have their advantages and disadvantages. If you must, you can use an even more complex system, such as insect cells. There is a very successful combination of baculovirus and Spodoptera and other insect cells that is slightly more complicated to grow and to maintain than yeasts and fungi, but not as difficult as mammalian cells, which is your last choice, and transgenic animals. These are the most complicated, most expensive systems, and you only really use them if you have no other choice. A big portion of commercial recombinant protein that is not produced by E. coli is produced with the baker's yeast or Saccharomyces cerevisiae. This is perhaps the best, or it is in fact the best studied eukaryotic cell that there is, and there is more genetic tools for this organism than for any other eukaryote. An example of a regulated promoter that we need for the same reason as for the E. coli case is GAL, the GAL promoters. We add galactose, and these promoters are induced, and we can have higher transcription. It's not as on-off, as absolute as the E. coli case, but we still have regulation here. It's used industrially. A notable example is insulin. Most or all recombinant insulin on the market is produced with the Saccharomyces cerevisiae. It's very robust, perhaps one of the most robust yeasts, and this means that it can tolerate low pH, high sugar concentration, non-sterile conditions, etc. A negative aspect of Saccharomyces is hyperglycosylation. That is, it has a tendency to put sugar molecules, chains of sugar molecules on secreted proteins that are not compatible with the human immune system, for example. As an example of that, we have Saccharomyces cerevisiae, and for secreted proteins, I should say, it's common for all organisms, all the eukaryotes really, to put sugar molecule to increase stability and effectiveness of secreted proteins. This does not concern proteins that are intracellular. They are rarely glycosylated. But Saccharomyces cerevisiae has a tendency to put big chains, and these are very, very irritating for the human immune system, for instance. So you really can't have these molecules. There is an example here of human, much shorter and also a different pattern. The Pichia pastoris yeast has a much smaller pattern of glycosylation, and this could be more acceptable. It's not perfect, but it's a lot better than the Saccharomyces. So this could be an alternative. Pichia pastoris is also a yeast used for other reasons. It can secrete and produce high amounts of protein. The yeast Pichia pastoris is a methyltroph, which means that it can consume methanol, which Saccharomyces cerevisiae cannot. Methanol is a very inexpensive carbon source, less expensive than sugars. We can also grow this organism to a very high cell density, and there is a pair of very strong promoters, AOX, these AOX 1 and 2, that control the alcohol oxidase gene that is used by the cell in the metabolism of methanol. We can replace, or we can use one of these promoters to transcribe our own gene, and we can get a very high protein production. There's an example of 12 grams per liter of a recombinant protein example here. The negative aspect, although it's positive compared to Saccharomyces, it's still not perfect. So the glycosylation is put down here as a negative point. Aspergillus nidolensis and Trichoderma rhizae are two filamentous fungi that are also used in biotechnology. The main advantage of these two organisms is that they can secrete enormous amounts of protein. This depends on their lifestyle. The Aspergillus you get on your bread. If you leave it out too long or leave it in the bag for too long, you will get this mold on the bread, and it's often Aspergillus. Trichoderma is a wood consuming fungi, so you will see it growing on wood in the forest. This lifestyle requires lots and lots of hydrolytic enzymes being secreted, and they can secrete hundreds of grams per liter of protein, which are enormous quantities. Aspergillus has a glycoamylase promoter that is regulatable by adding starch or xylose to turn on and off the gene, which can be very useful. There is an Aspergillus niger that is used very much in. It's closely related to this Aspergillus nidolensis, and is widely used for citric acid production. There is a cellulobiohydrolase promoter in Trichoderma that is turned on by cellulose, which you can understand from the lifestyle here of these fungi. And we can use addition or the presence or absence of cellulose to regulate the transcription. These organisms tend to be very robust, and they tend to be happy with low-cost substrates. The Aspergillus niger, for instance, is used industrially for citric acid, as I mentioned, and the final titer can be several hundred grams per liter of citric acid, and this is a prohibitive concentration for most organisms, but Aspergillus seems to tolerate this. If we still cannot use fungi, perhaps because of the issue of glycosylation of the proteins, we can step one notch up and use insect cells. These are essentially cell lines or cancer cells isolated from a moth called Spodoptera frugiperda. So this is a cell line is of course a cancer cell line that divides indefinitely, and we can use this as a base for protein production. We have to modify it, of course, in order to produce our protein, but it can be used. The advantage of this system, as compared to mammalian systems that we will talk about later, is that it's still quite uncomplicated culture. We can culture these insect cells, although they are mammalian cells. We can culture them in shake flasks, just like we do for yeasts or E. coli, and we don't need to control the atmosphere too much. What this means, I will come to what this means when we talk about the mammalian cells. But the message here is that it's almost as easy or inexpensive to culture these cells compared as for E. coli or yeast. It is a little bit more tricky to clone, to get your gene inside. It's a bit more complex because there are no plasmids for these cells. We need to use a virus. The vector of choice for insect cells is called baculovirus. This is a virus that can infect only invertebrates like insects and other, and it has an interesting infection cycle. While inside the insect, it can infect other in cells within the same insect by producing and secreting this structure here. It's the virus inside. It's it's a double-stranded DNA covered with a positively charged protein, and it has a membrane here derived from the cell membrane of the insect, and it has a membrane protein that facilitates infection of other cells. In the later stages of the infection, these occlusion bodies are produced. That is actually a big crystal structure, and you can see one here in the microscope image. And it consists of a polyhedrin, crystallized protein, and these viruses, particles here packaged in different ways. The point here is that large amounts of this polyhedrin protein is needed to make these structures. So there's a very strong capacity for production, strong promoter, etc., for producing this protein. And we can replace, it's not an essential protein, so we can replace the polyhedrin gene with the gene for our favorite protein and get lots and lots of our target protein instead. Cloning with baculovirus, as I mentioned before, is not as straightforward as for normal E. coli, for example. Since the genome of the virus is very large, it can be around 100 or more thousand kVs. This is kind of difficult to manipulate in vitro. So what is done is that you separate most of the genome of the baculovirus and your gene of interest, and these two molecules are then combined there are different ways to combine them. The original protocol called for co-transformation with the plasmid that you have your gene and the big genome of the baculovirus, and these would recombine in vitro. The problem of this was that it's very low frequency of recombination, maybe only 1%. So you had to look very hard for a clone that contained the correct baculovirus. There are modern variations of this example here where you recombine in vitro using a recombinase. So this gene here of your interest, the one you want to express, can be recombined with an enzyme in a test tube with your backbone. So this DNA can directly be used to infect insect cells. Even when you have your infected cells, you have to obtain enough virus to get a reasonable amount of protein in the end. The baculovirus is not lysogenic, it only has a lytic cycle. So you need to grow in several stages in order to obtain more and more of the virus, in order to make a final culture where you use all of the virus to infect as many cells as you can in order to get the amount of protein that you want. Finally, you have mammalian cells, which is really the most expensive option that can be used for production of proteins. And it's really only used for important proteins, such as therapeutic proteins or proteins that are going to be injected into the bloodstream of people. You use usually, since these are high-value products, you use these kind of advanced equipments for a very high level of cleanliness and control. This is a typical smaller scale reactor than this one, and here you have to control temperature and other variables very carefully. The atmosphere needs to have enough oxygen, otherwise the cells die. You need to control pH very, very carefully, and you need at the same time limit the shearing forces. That means that you can't stir this reactor as much as you want. You can mix as violently as you want. With E. coli and a yeast and fungi, you can stir as much as you want, really. But here you have to be careful, because otherwise the cells die. And many cells actually need a physical support. These are cells that are used to being in the body of animals, and they need other cells in order to feel good. What has been done is to develop these small supports. This is really a round bead, and you can see that it's covered. Each of these things here is a cell. It's some kind of human or animal cell, and they grow as much as they can here on the surface. If they fall off, they die. So you need to carefully select these supports and then find a way to still transfer enough oxygen for the cells to grow. Another problem with human cells is that they are quite similar to ourselves. So human and other mammalian cells, they are very similar to us, which is good, but that's also a problem. They can be infected by the same kinds of viruses and other diseases that infect human beings. One problem in association with this is the use of fetal bovine serum. So this is a solution containing many nutrients that are needed more or less for cultivating human and other mammalian cells. And this is not a very, it's produced not a very nice way. This is actually the blood of unborn fetuses from cows that are pregnant when they are slaughtered. So the fetus is extracted and its blood is extracted, and then the red blood cells are centrifuged away, and this is packaged. One of these bottles costs in excess of 500 euros, so it's a very expensive additive. And this contains many growth factors and other compounds that are needed for cells that we don't really know about. It's not a defined solution. And this means that we cannot really guarantee that there is not some kind of virus that sneaked into this bottle from the slaughtered cow. although the fact that we use fetus for this is a safety measure. There's no antibodies in this, for instance, in this solution, or ideally no antibody. It's not a very good proposition. It's to have this uncertainty. Nowadays, I should say that there are cell lines that don't require any serum, and this is preferred, of course. It also lowers the cost of the product. In this context, we also need to mention the production of monoclonal antibodies, which is a slightly different form of cloning, really. We start with a cell, antibody-producing cell, and we fuse this antibody-producing cell with a tumor cell. This is a cell line here. And by a small frequency, we will get cells that are both immortal, like the tumor cells, and easy to cultivate, and they produce antibodies. So here, instead of extracting whatever gene that we have in this antibody-forming cell, we fuse it with another cell in order to obtain a cell that has both advantages. This cell here is impossible to cultivate. It will die soon after we extract it. And these cells can be grown as any of the mammalian cells that I've mentioned before. You usually have to screen for cells that produce a lot of antibodies that you want. There are, of course, attempts now to extract some of the genes that encode for the interesting part of the antibody here and express it in E. coli or in yeast or in some other more simple organism, because in the end of this scheme here, you still have to grow this mammalian cell here with all the costs and difficulties that involves. Finally, the most complex system must be recombinant animals, like sheep, for instance, that could produce recombinant protein in the milk. It's very costly and expensive to make these animals, but once you have them, they're not very difficult to maintain. Most of these animals are actually made in 2 steps. You have a DNA solution, and this DNA solution contains the gene that you want to express. You inject it in a somatic cell. This could be any cell from the animal. You do this because for most animals you cannot directly inject into an egg cell. The egg cell will not accept that. But the somatic cell, for instance a skin cell, could be used to any tissue cell. You remove the nucleus and you combine the egg cell without its nucleus with a new nucleus. And then you have to apply a lot of engineering in order to reprogram this nucleus so that it can start and divide into a new animal. If you are lucky, then you will get an animal that has a good production of, if you put this gene in the right place, for instance, in the glands of the milk production of the sheep, for instance. The problem here is that the animal has a limited lifespan, and it's really a very expensive undertaking to create an animal. Also, you have to test many animals in order to get a good producer, an efficient animal that produces a high titer of your protein. This was the main motivation for animal cloning. You might remember the Dolly the sheep that was created many years ago. The reason for carrying out that research was that when you had a good sheep that produced milk with your nice recombinant protein, you could in theory clone that sheep and get as many clones as you want, and in that way you would save your investment in the beginning, and you could get a much higher production capacity. In the end of the day, it comes down to choice. This figure here tries to compile the advantages and disadvantages of the systems that we have discussed. The BEVS is the baculovirus that I mentioned before. You could say that there are now hundreds of approved therapeutic proteins that are approved by the Food and Drug Administration, and they can be part of medication. If we break down this by system of their production, about 20% are produced in yeasts, and this is mostly Saccharomyces cerevisiae. About as much or a bit more, 30% in E. coli, and the rest in mammalian cell lines and hybridomas. So even though the mammalian cells have all the disadvantages that I mentioned, it's still a popular choice, mostly because they are guaranteed to express human proteins, roughly. So here, if we look at the details of this, we can see that the bacteria is the fastest system. What they aim at here is speed of getting your production going. The cost, the bacteria is also the cheapest. Yeast here is almost as fast and almost as cheap as bacteria. The glycosylation is very bad for these inexpensive systems, and this is probably the main reason for the choice often of the mammalian system. Folding also, this is how efficiently the system you use are able to fold your protein correctly. Again, mammalian cells come out the top. And then again, how easy or hard is it to get your system through to be approved? Here, mammalian yeast and bacteria group together because of this reason here below. They have been used in many systems, and it's quite difficult and expensive probably to convince the FDA that you should use a new system when these have been studied thoroughly.